see. And thanks to Dave for having me here. Oh, should I?
1 second. I'm gonna just.
Oh, we're recording. Yeah. Okay. All right. Here we go. Good call. All right. So I think probably latent factor discovery is an idea that comes up for a lot of you, and I'll give you a little bit of our motivation or how we think about this problem. This way. Move the mouse right here instead. Oh, what work do you click on this?
From that desktop.
Okay.
And you could try now.
Okay. Yeah. Okay. Well, now it works. Okay. So I'm going to start with like a little science experiment that you've all done many times, where you enter a room and then you turn the light switch on and you establish a causal relationship in your mind anyway between this light switch and turning on the light in the room. So one reason that this is like a very when you get into the situation, you probably don't do a randomized control study for yourself to like really feel confident about this. You just flip the switch once and you feel pretty good about it. You don't need a lot of statistics. You don't need a meta study of 50 studies to establish this. And so there's a few reasons why that's the case. I mean, obviously the effect size here is extremely large, right? So you can immediately see that there's been a big change. But the other thing is that your knowledge about the dimensionality of the system, which might be relevant, is also very small. So for instance, imagine that somebody invented a new sequencing technology, and we were going to put it to use in this experiment, and we would be able to measure the state of every atom in this room. And we'd generate some huge data set like this. And we would write a bunch of NIH proposals, and we'd say, we will get unprecedented understanding of the light physics in this room or whatever by using this next generation sequencing technology, because we have way more data and more is more, right? But actually, I mean, I think you probably can see that in this case, we would actually be much worse off, right? Because if you look at this data set of the state of every atom in this room, you'll find many of these atoms or whatever whose state is perfectly correlated with the light in the room, but they're totally useless. And if you took one of these correlations and published it and then said, we're going to design an experiment that knocks out this atom, you'd see that you get nothing from that, right? This is like not a good thing to do. So the issue here is that really it's very difficult to go from this microscopic view, which gives us more data, but really we somehow like to map from this microscopic data back to these, what I would call macro states. Like in this case, it's the light switch, right? So how do we get from the microscopic data to the light switch data? And so I view a lot of things from that point of view, like you could say human behavior or something, where you can see a lot of like individual decisions that a person makes, but we don't actually care about like every single choice that you made. We care about some sort of pattern that represents your overall state. And gene expression is something that's the same way. It's a ton of data, but we don't really care about the exact expression level of every gene, which we actually think is extremely noisy. But we think there should be a way to recover these sort of high-level macro states from this data. So this is our kind of motivation, I guess, is to do this. And if we can identify macro states, we also hope that it will be more useful for making predictions about outcomes for different sorts of interventions. So that's the goal, or the kind of framing or motivation for what we're going to talk about. So we're doing a joint talk. Myrl's a PhD student in my group, and so I'll describe one way in the past that we've approached this sort of macro state problem, and then some of the problems, and Myrl will present the solutions to this problem. So in the past, the way that we worked on this is using an information theoretic point of view. So the information theoretic point of view is kind of summarized with this, which is we have these high-dimensional variables, and we want to construct latent factors which are maximally informative about the data. Now, there's actually a lot of ways that you can characterize what you mean by maximally informative. So in our case, what we're saying is that mutual information is a general way to measure relationships between variables. There's a multi-variable extension of mutual information. Some people just say multivariate mutual information. Some people call that total correlation, which is kind of a misnomer. It's not a correlation, it's a dependence measure. So the way we measure this is to say we would like to find latent factors such that the total correlation among groups of variable is minimized conditioned on the latent factor. So that might not sound very good. What I'm saying is that these variables have very strong relationships, but if you know the latent factor, you can completely explain those relationships. Conditioned on the latent factor, you know, everything factors very nicely. And so that's very simple. Everybody has an umbrella. You say umbrellas are very correlated, but conditioned on whether it's raining, that explains all the correlation. So that's what we're trying to do in an information theoretic way. Construct the latent factors that capture the maximum information from this point of view. Okay, so I have sorry, here's my my animation of this, which got a little bit messed up. So the idea is that we're going to have some high-dimensional data. Again, that's these columns. Maybe this is gene expression data. We have a small number of samples of different people. And you know, your data matrix might look something like this. So the first step, kind of algorithmically what's happening, is to say, well, we have to rearrange it and find the groups that have high multivariate mutual information. So that's what this sort of reorganization is doing. And now, associated with each of these, we're going to reconstruct a latent factor that would make these conditionally independent. So, for instance, the way that's done is sort of to combine the information in some way through an optimization. And one of the advantages of looking for these maximally informative latent factors is that the reconstructed latent factor can be more robust than any of the individual variables that you looked at, right? So that's kind of the goal, is that we have many noisy variables which are correlated, but we want to turn them into a small number of these macro state variables which are more reliable and tell us about some phenomena we care about. So that's the basic idea. And just to give you a little sense of like, okay, so there's lots of mathematical ways we could approach this. And then the hard work is really doing something with it. And I think that's where people in this room come in. Like, this is the hard part by far. So it's always hard with these unsupervised methods to figure out whether what you discovered is really useful or not. And so most of our effort really goes into that. So in this case, I said we're maximizing some sort of information measure. And in this RNA-seq study, we get some sort of like map of latent factors over some thousands of gene expression variables. And when you look at any one of those, you see these kind of plots, which are probably not unfamiliar to you, where you have a group of genes and you look at the expression levels, and you can say, you know, this makes sense, right? That this group of genes has high multivariate mutual information, and you can see that even just by glancing at it, that these are all very highly correlated genes. So then what we really want to see is whether there's something interesting about these kind of discovered latent factors, these discovered groups. And the way that we often do that in practice is not perfect, but the point of view is that if you really are good at discovering latent factors in biological data, a lot of what you recover should be known biology. Like, we already do know a lot about biology. So the way we test this is to say, you should at least be better at recovering known biology than other methods. And then the hope is that if you're better at discovering known biology, you can then look at the sort of extra variables that we don't know about and see whether these are really something that is unknown and that's been discovered. So the way we often do that is this type of enrichment, gene ontology enrichment, where we say, okay, this group of variables has high mutual information. If we put it into one of these databases, we'll see that this group of genes is highly enriched for a specific functionality. And then we just want to compare these to other ways that we could have done this clustering. And we see that indeed we can get much more sort of enrichment of known biology, suggesting, you know, we're moving in the right path to discovering known biology using these methods. And then the last thing is that, of course, we really would like these methods to, first of all, reflect biology more faithfully, but also that we can use these latent factors that we construct as more robust predictors of the things that we care about. So the last kind of way that we test these methods is by seeing whether the discovered factors are better. Are they more robust at predicting outcomes? So again, this is not perfect because a lot of really interesting biology is not related to the outcome that you might have in your data set, but it's one of the few things that we can do. And so this is an example from that study where we see that, we can see huge differences in one of these factors for 5-year survival. And that factor is very associated with very specific targeted biology, which gives us kind of something that we can work with. So this is sort of a way that we've been looking at these, at the gene expression problem, recovering latent factors. This study was all done with bulk RNA sequencing. And, it's a tissue, it's a sample from tissue, and it includes a bunch of different cells. In a way that can be good because you're averaging a lot of things. It makes the signal like closer to an average signal. But we have even sort of more precise view now in single cell RNA sequencing, but it's much noisier also. And so with this much noisier data, you can actually have a problem that it's very difficult to discover these relationships in the way that I presented here. So we really want ways to deal with this, more fine-grained, granular data, but still recover something very robust from it. So this is something that Myrl's been working on a lot lately and has some interesting results. So I'm gonna turn it over to him at this point. Here you go.
Thanks, Greg. Very excited to be here. So, as Greg mentioned, I'm going to be discussing a new method we've been working on called q-Diffusion, and it's been built specifically for the challenges present in single-cell RNA sequencing, or scRNAseq to abbreviate it. And in particular, our method is supposed to be able to use all the dimensions available in scRNAseq, as opposed to existing methods that throw away a lot of the information with PCA or dimensionality reduction, things like that. So first, a bit of motivation. I think it hardly needs mentioning that scRNAseq it has a lot of potential. It's been the Nature Method of the Year multiple times now, and we're witnessing the emergence of atlases with millions of cells for specific conditions like metastatic colon cancer and other things. And we're also seeing multimodal data come out where you have transcriptomics, and then also on top of it, you record like electrophysiological properties of cells in the same cell, and then you could associate these things. And that's pretty exciting. What I want to talk about is how to analyze transcriptomics and how to come up with images like these, and perhaps better images where perhaps you've seen these pretty often. These are where you plot all the different cells according to how similar they are to each other. And then we hope to see these groups separating that describe maybe phenotypes, macro phenotypes. So briefly, these new opportunities that are afforded to us by the more granular data also invite new challenges. There are some traditional difficulties with biological data sets like batch effects and whatnot, and these are only amplified when we go to the granular level of a single cell. It seems, for instance, that there's a trade-off between batch effect removal and preserving biological variation. In other words, you'll always lose some real information if you try to remove all the batch effects. And not only that, that's just an example. There's also like strong separation between things that we don't want to separate between just individuals as opposed to the disease state, for instance. So very generally, the dimensionality tends to be too great for complex statistical methods, and we have to rely on simpler things that work with that are more sample efficient. And just briefly, the way that we analyze scRNAseq is that we look at cells as a very high dimensional vector of gene expressions. And then a natural question that we try to answer is, what space do these live in? What's the geometry of these high dimensional vectors so that we can compare them and get structure out of them? So what makes two cells similar, I guess, is a simple way of trying to answer this question. Like, well, for a pair of cells, what makes them similar? What makes them different? We answer that with a kernel function, as we call it. Now, in typical methods, we use something called the Gaussian kernel, as you've probably seen. Visualizations like these tend to use Euclidean distances between vectors, but only along a small number of dimensions. And that's because of the curse of dimensionality, as I'll elaborate on later. But I'd like to look at two perspectives of our method q-Diffusion for how it addresses these broad challenges in single-cell RNA-seq. The first is biological variation. So when we look at all these different phenotypes in cells, especially when we want realistic data from humans in the real world and all that. There are a lot of competing processes that are interfering and affecting individual genes. So for this reason, as Greg mentioned, we want to look at latent factor models to hopefully disentangle these processes. So we could look at individual biological processes on their own and describe the cells at the proper level of abstraction, harkening back to the light bulb example I like a lot that Greg was mentioning. So very generally too, we want to be able to capture the emergent properties of biological systems, not individual genes, but what emerges as a higher order effect of these genes interacting. So this is a pretty abstract idea, and I didn't know what to put for a figure here. So I asked an AI to draw one for me. So I asked, draw me emergent properties of biological systems. And here's what it could say. I'll leave the interpretation up to you. Some tomatoes, this looks like an egg, maybe, I don't know. Anyways, so this is the core of q-Diffusion. It's a geometry of gene co-expression. So, as we mentioned, biological processes comprise genes that tend to express, regulate together, many groups of genes and large groups. So we constructed q-Diffusion, or the q-diffuse kernel, specifically to consider two cells to be different, not when individual genes have different activities, but when they're large groups that change together. And one natural thing to look at would be like pairwise correlations between genes. But we went further and we incorporated every order of interactions. So we have pairs, and we have triplets, and we have quadruplets, and in the cascade of diminishing importance, we have this alpha coefficient that is less than 1. But we're able to capture basically interactions that are across many, many genes. And we try to emphasize the bigger interactions more. And this is how we do that. Also, as a spoiler, the second perspective is a bit on noise as opposed to biological variation. And this construction of our distances plus the power law decay on our kernel give us this resilience to noise as well. So it's not just helpful for biological variation and latent factors, but it's also helpful for being resilient to noise. And they kind of go hand in hand. That's what's exciting about this method. But first, I'll kind of elaborate on the multiscale properties. So the curse of dimensionality could be interpreted as if you just need like 1 or 2 dimensions to really throw off your distance function because you're adding squared distances, right? Then as you get more dimensions, you're guaranteed to have high distances always. So everything is about as far away from everything else. That tends to happen if you just take square distances between really long vectors. With q-Diffusion and the thing I mentioned where we emphasize the large interactions, we see that the number of big differences in dimensions, that number of dimensions scaled with the total number of dimensions in the data. So in other words, if you have 20,000 dimensions, 20,000 genes, you need a lot of those genes to be for a big difference to register, for the kernel to decay from yellow to blue, which is from high to low. So this is just kind of showcasing how we sort of overcome the curse of dimensionality when it comes to these biological systems specifically. I mentioned this inner bandwidth thing. I think what we need to know for now is that we have a simple heuristic for computing it, but it's pretty important for q-Diffusion, and we can mention it if you're interested in later. So here's the first set of results that we got. We looked at PBMCs, which are pretty standard, peripheral blood mononuclear cells, and there's this really cool data set where they stimulated them with interferon gamma, and we want to be able to figure out for each of these types of PBMCs, like B cells, is it stimulated or not? And interferon gamma has a very multivariate effect on different cells. So traditional methods kind of struggle to separate cells based on this interferon gamma stimulation. q-Diffusion is able to do a lot better for the majority of the cells, B cells, CD4, dendritic, etc. And I think that points to the multivariateness of these separations and how we need to use things like q-Diffusion for that. So it's pretty exciting.
Sorry to interrupt, but the optimal transport? What is that?
So that's a new method. I don't know if you've seen in very recent papers, some people are introducing it. It's this way of comparing cells according to so let's say there are a few genes that differ for some cells. You look at how different those genes are, and you kind of say, okay, if the genes are very similar, then even though they have a big difference, the actual difference between the cells is not that big. So it's trying to compute distances between cells using distances between genes through like ontology structures or things like that. So yeah, I'll mention it a little bit later too, but a lot of questions remain to be answered in how you implement optimal transport, but it's another direction, yeah, for these things. It does work better in these two cases, so that's something. Yeah.
One sort of a specific question. Are you comparing to a ground truth here? Like,
yeah, so we know.
How is that different?
With sample multiplexing. So there's this method, I forget who did it, but you're able to multiplex batches if you knew you have like genetic markers that you could apply to cells. So even though you could have different groups, you could put them in the same batch and then still know which cells belong to the two groups. But I think it's difficult to do because most people don't do that. So instead they rely on probabilistic, sorry, maybe it's too far. Anyways, oh wait, I'm going way too ahead. Maybe it's probabilistic methods that that we need to use to do multiplexing, but they don't work very well. So it's a rare data set to have that doesn't have batch effects, basically. All right. Technical noise is the second perspective. There's a bit of a controversy in whether single-cell RNA sequencing is zero inflated, but we do know, regardless, that it's very noisy, that there are a lot of zeros because we just don't happen to read a lot of expressed genes. And perhaps counterintuitively, we find that the multiscale property makes us more resilient to this technical noise, because imagine if you kind of zoom out, you don't care anymore about individual genes dropping off because you care about kind of the bigger picture. And in particular, this technical noise is mostly uncorrelated. So even if there's heavy noise, because it's uncorrelated, we don't care about it anymore. We care about the big interactions, and it doesn't really affect us as much. Here's just the cascade of steps in which gene expressions that really happened might be lost in the data. So there are a lot of stages in the pipeline in which this could happen. We know it's very noisy. We did an experiment where we have two phenotypes. This is simulated, it's synthetic, but we have two phenotypes of different structures. We could have like the two phenotypes differ with just a few strong genes or many weak genes. And then we could also have different levels of noise, so different amount of zero, not inflation, but a lot of zeros being thrown in. And we basically found that q-Diffusion does best when you have a lot of weak genes separating phenotypes. And even with very high noise, it doesn't care. So that's pretty interesting. Even here, you see that like when you have phenotypes that are separated by just a few, strongly acting genes, more noise makes q-Diffusion work better, strangely. But we have some hypotheses for why that might be the case. It's kind of the zooming out thing. It forces you to not look at the individual genes, but look at the bigger, fuzzier picture kind of. And then I just find the kernels here. So this is bivariate. We have imagine you just have two genes, right? And how much do the genes need to change from zero to make the kernel decay, to make cells look different? The Gaussian kernel is just a sphere. It has these like concentric circles. Then the q-diffuse kernel becomes kind of concave. And that's kind of the magic in the end of what our kernel does. The kernel only decays when you have both dimensions increasing at the same time. So not just horizontal or vertical, but you have to have the diagonal. And that, in the very high dimensional setting, becomes a lot more interesting than this, and it also has other properties. So what we're excited about with q-Diffusion especially is that it could augment many common algorithms. So we don't have to invent new ones. We could just use things already like t-SNE, non-negative matrix factorization, and others. So with scRNAseq, one thing that people tend to look at is gene expression programs. They want to look for the set of genes that express together and decompose cells into these programs. NMF is a way to factorize cells linearly so that we look for genes that express together, not regulation, but just expressing. And it seems to help. For instance, if we do clustering on the basis of the gene expression programs, the weights on these programs rather than the genes themselves, we get the cluster phenotypes a lot more coherently than if it's just on the gene space. But it's linear, so we lose, it's not the most sophisticated method, I guess, for getting these structures out of the cells. So one thing we found is that if we put a q-diffuse kernel on top of all these cells, and we found their similarities in this q-diffuse space, then And then we force the gene expression programs to kind of obey this q-diffused constraint, we get much better gene expression programs, GEPs. For instance, looking at enrichment, like Greg did earlier, we find that enrichment increases for Q and MF, which is our new thing, compared to NMF and all the competing methods as well. So that means that there's additional information, the structure that is conferred to us by q-diffusion, and putting that into a linear system like NMF still makes things more coherent. All right, so now the big case study that I want to talk about is metastatic colon cancer. We've been working with Dr. Heinz-Josef Lenz's group, and we're grateful to have access to this data set, which is a clinical trial that contains bulk RNA sequences of patient tumors. And they've been given two different treatments, two different randomized treatments, bevacizumab or cetuximab. These are the two frontline therapies that are used for metastatic colon cancer. And they've already been able to find all kinds of things that are used in international guidelines these days, but we still need a predictive biomarker for bevacizumab. That's something that hasn't been found yet. And we constructed this little case study where we thought, okay, could we look at a colon cancer scRNAseq atlas, get the most informative gene expression programs out of it, and then transfer it to these patients who are bulk RNA, but we have, treatment survival information, all that other stuff. So if we transfer there these same latent variables, and then see if there are differential effects of these latent variables in the patients on their survival, right? And here's an outline of this. We took the atlas, we got these two versions of latent variables, the gene expression programs I've explained, branching gene components I'm going to explain shortly in a few slides, but they're just two ways of decomposing the cells into these latent variables, and they become linear in the end of the day. So then we're able to transfer them onto the bulk RNA-seq patients, and we do a cost regression on survival, and we look for an interaction. And what we found is that both of these did give us some strong differential effects. So a latent variable that if you find it in a patient, it tells you if they're going to do better with bevacizumab compared to cetuximab in a statistical significant way. For the gene expression programs, we found one that has with an FDR less than 0.05, with q-diffusion. Without q-diffusion, it's not significant. And then with this other thing called branching gene components, we also found something strong, FDR less than 0.1, but without q-diffusion, there's nothing, even that. So with these two groups, we found latent variables that have differential effects. And then to interpret these latent variables, we found the individual genes that also have strong effects on bevacizumab outcomes. And all of these genes, it seems, are novel in the literature, except for E-rate. But all the others are novel in their effect with bevacizumab. So it's suggestive of novel biomarkers. And then, real briefly, the branching gene components thing is we took, if you're familiar with UMAP-T-SNE, there's also another thing called FATE, which is an embedding method, so a dimensionality reduction method on cells that looks for structural embeddings, things that are not just for clusters, but maybe it can tell us something about the structure of the data set. And this is of the whole tumor microenvironment, right? So we did a q-diffused version of FATE, and we found this structure that has two major branches and one smaller branch. And we thought, okay, maybe the genes that differ the most between these two branches will be the things that might drive the branching structure in these microenvironments. So we took the genes that are most differentially expressed between the branches, and we used those to find the other set of latent variables. And there's some interesting stuff in there. So that's another way to look at it. But here we've augmented a common embedding method, FATE, with q-diffusion, and it gave us more interesting things. It gave us branches, whereas the old version does not. So that's something. So also, scRNAseq analysis, I want to talk a little bit about how we compare it to the state of the art, how people do analyses. I just want to mention that for typical state of the art pipelines like Seurat and for Monocle, those things, you need to do a bunch of pre-processing first, like aggressive feature selection. And after feature selection, people also standardize sometimes. They make OS standard deviations the same because they want to emphasize the quiet genes like transcription factors that are very important, right? But it's kind of weird also because you lose a lot of information by first removing most of the genes and then standardizing the rest. And also on top of that, we tend to do dimensionality reduction like PCA to get like 16 dimensions. And then you do everything on top of that. You do the embeddings, you do clustering and whatnot through the neighborhood graph that we construct using the Euclidean distances on just like 16 dimensions. So we go from 20,000 to 1,000 with feature selection and then to 16 with PCA. And we need to do that because it's noisy, but q-Diffusion, it obviates all these steps. So we kind of use the full dimensionality to characterize our cells. Now, as was mentioned, optimal transport is another interesting direction, but yeah, as we said, there are some operational questions that need to be answered. Like, what does it mean for genes to be similar or not? We're not just looking now at cell similarity, we're also looking at gene similarity. And there are many ways to do that, and we're not sure what the best one is yet. And here are how the different methods compare, right? Seurat, ScanPy, SC3 monocle, optimal transport. The main takeaway is that we don't have the pre-processing steps really, except for the log normalization that everyone does. We look at all the genes, and yeah, it's also simpler in many ways. So for next steps, I want to emphasize that q-Diffusion could help inform other larger machine learning pipelines in the future. Because even though, with deep neural networks, we say, okay, we just throw a neural network on it, we see what it's able to generate, see if it can find its own answers to everything. Still, the best methods need to have some information about the structure of the genes. Like we either look at neighborhood graphs or we use kernels to regularize the models. So we could just as easily use q-Diffusion to regularize the big generative models that are making all the headlines these days. And I want to work on bridging the gap between advances in causal inference that the machine learning community has made just in their own bubble of standardized benchmarks and clean causal problems, and scRNAseq analyses, which are where the interesting things are happening in life. And maybe this, right, because if you could do causal inference on scRNAseq, you could try to simulate interventions and maybe do things like potential drug target discovery. And that's what I'm looking at currently, or hoping to at least. So thank you very much. I want to thank my collaborators too. And I'll end on this slide. Thanks.
Okay. Any questions? You here? Let me check online. Any questions online?
So how do you implement this? Do you have a package?
Yeah, we have the GitHub package, but it's implemented in Julia, this language we like, and we've implemented also CUDA, so GPU code, so it could run and parallelize and be very fast, because otherwise it's pretty slow to compare all these cells. So yeah, we have all the code here, and it's still a little early in development, but feel free to reach out if you want to use it. I'm happy to help with any use case. Yeah.
We do have a question online. Nada?
Hey, Myrl, it's actually me. It's Paul Thompson here. Sorry, I was overhearing your talk and it was really cool. I had a question about the form.
Hold on 1 second.
Oh.
Wait.
Oh.
Okay, try now.
Sorry, can you hear me okay now?
Try again.
Can you hear me okay now or not really?
Yeah.
Can you hear me okay now?
Well,
Sorry, can you hear me okay now?
Yeah, why don't you go ahead and
we can try it. Yeah.
we'll see if we can repeat the question. We all get it as a group. An
Okay.
aggregated listening experiment.
Okay. How about I chat the question and then Jose Luis can ask one?
Oh, okay. Okay. Excellent. Jose Luis.
Paul Thompson and Nether together.
Yeah.
Well, I'm also remote. I mean, I hear Paul perfectly well, but if you're gonna keep me in the room, so I have like a kind of technical question, but more the intuition. So the q-Diffusion has this parameter q or alpha.
So it's hard to hear you. We're trying to figure out the volume still.
Okay.
Try again, Jose Luis.
Can you hear me now?
No, I don't think so.
Can you hear me? No.
Just when you think you have all the technical
Yeah.
things handled. Maybe Jose Luis, if you could write it in the chat,
I'll test it. Yeah, I'll chat it.
while we're getting the chat to questions to come in. Any?
Yes, sir. Yeah.
I just want to sort of, like, I think I'm understanding, but I'm not sure. So when you say that you're using like q-Diffusion for clustering, you're still using like some underlying clustering method, like moving or lighting. But yes, there are differences the space in which you do it. You're
exactly.
On your factors.
Yeah. Okay. So if you're familiar with like how like Seurat does it, you do PCA, all that kind of stuff. You get distances between cells. Then you build a neighbors graph, and then you do commutation on the neighbor graph.
Right.
So we build the neighbor graph with q-Diffusion instead, and then we do those same commutation algorithms on top of it.
Okay.
Yeah.
How does that relate then to dimensionality reduction visualization?
So these are different. So we could do q-Diffusion on all of these, right? So you could do it for clustering or dimensionality reduction, or for we could do like q-Diffusion as well, for instance. So yeah.
Like how normal, like in like the traditional way they do PCA and then TSNE, you would do.
Yeah, exactly.
Yeah.
Exactly. Yeah. Yes, and also NMFs. Yeah.
Yeah.
Cool. Great. So we have so a couple questions here. Jose Luis is asking, what's the effect of Q alpha? So how do you tune the hyperparameter? Oh yes, that's a great question. So yeah, I showed alpha and Q. Alpha is related to Q, just like Q minus 1, I believe. Q greater than 1 is, so Q equals 1 is the Gaussian kernel. So, and then we just set Q a little bit greater than 1 to get our interesting Q diffused effects. For all of our studies, we've just done Q equals 1.2. And we just look at that for now. It's an easy quantity. Perhaps in the future, we should look at changing it, but that's it basically. Q equals 1.2 corresponds to an alpha of 0.2. Yeah, and it just, we have to just, honestly, there's not that much of a difference now with changing the Qs. We just need something greater than 1. Yeah, and that's it.
I guess the question, you're gonna hear me, but the question is like,
the additional one, is there a good default? And if you discount too much, do you miss the high level interactions?
Yes.
And that's also related, I think, to the question from Paul Thompson. In the q-Diffusion formula, there are lots of nth order interactions. You showed it going out in a mini. Do you mainly detect second order interactions, and are they dominant? And do you need more training data to estimate the higher order interactions?
Those are great questions. So first of all, I guess for the discounting, if you discount a lot, you end up losing the higher order stuff, and you only look at the low order things. Also, if you don't discount enough, it just gets a little too wacky because you really emphasize the high order things. So it has to do with the dimensionality, for instance. If you have a really high dimensionality, you don't need Q to be that high before things start changing a lot. Yeah, so it really depends. Basically, the higher the dimensionality, the more effect Q has, so you don't have to make it that big for it to have an effect. Now, the other part of the question was looking at just second order versus higher order effects. I think we could say that, for instance, PCA looks just at second order effects. So the fact that we get vastly different results from PCA means that our higher order parts do mean something as well. And I guess that's the way of answering the question.
I guess a follow-up question, and a couple follow-up questions. One is just understanding then, not just the clusters, but the components to the q-Diffusion process to better understand what are the higher order interactions and what would be driving those. Is there a way to do that? So it seems a little bit box right now. It's being a kernel. It's like often are, but
it is that is a great question, and we've tried to shed light on it by, for instance, doing these factorizations like NMF guided by q-Diffusion. So then what comes out of NMF is what q-Diffusion has found to be the important programs that exist inside a cell, right? Because q-Diffusion itself, is super just unsupervised. We have this one form, but what comes out of it downstream does inform what q-Diffusion has been able to find in a way. So that's how we've interpreted it so far.
And I think that's an important question related to the last part, I think, of Paul Thompson's question of the size of the training data. nothing's for free.
Exactly.
And it seems like you're balancing between linear combinations in the Gaussian kernel with this Q, or shifting it more towards higher order interactions, guided by not just the dimensionality, but also what's going on underneath is the power for detection of higher order interaction, right? I mean, you need a sample size that actually can allow for that. So what is, I mean, you showed an example with 1200 in Heinz-Joseph's data, but like,
we've gone from, for single cell, we've gone from like 5,000 to 100,000, and it's worked very well for all of those. With Heinz-Joseph, it goes to, we have like 1,000 patients, but we find the programs in the large atlas, which is I think 50,000 cells after doing some selecting. We find those programs and then we transfer them onto the fewer patients. So the actual discovery is happening in the larger sample size. But yeah, so we've tested it on a few samples, and I don't see an issue. It's not, I don't think it's that higher order interactions necessarily need more data, because in a way the kernel itself is power law and stuff, and it's able to find similarities between cells even if it's kind of sparse. Compared to the Gaussian, sometimes it struggles. It's huge, but the sample is tiny. Then the Gaussian kernel really doesn't find anything. And in a way, the q-diffused distance function that we have is able to, it really helps, I think, in many cases for that.
Wouldn't it be overfitting the data then? And so you need, I mean, I know you have a training and testing, but really the ultimate goal would be to have sort of an additional data set in the Heinz-Joseph case of looking at those genes and seeing if they really do.
If they matter? Yeah. Well, in a way, Heinz-Joseph's is a testing set, because we get our stuff, we train on an atlas, and then we look at the clinical trial and we test it there. And we see significant effects in the clinical trial data that we've learned in a different atlas. So yeah, and we also have, anyways, but yes. Good questions though, yeah.
Any other?
You mentioned at the beginning things like batch effects and the trade-off between, getting rid of that and getting rid of biological signal. Do you, it seems like, I don't know, I just want to revisit that, I guess. Do you pick those up in the q-diffusion dimension? Like, do you sort of do some pruning to say like, oh, this one's, patient level effect, or this one's a batch effect, or this one's...
You still pick them up, but they matter a little less. Because batch effects they're not really high order, I guess is the way of putting it. And we see, for instance, when you get clusters, they tend to be more mixed with different patients from different batches than other clusters. So, than like the Gaussian kernel, for instance. So it matters less, in other words, yeah.
Any other questions online in the room? If not, thank the speakers again.
Thank you.
Also, just for those here, there will be the usual sort of meeting after the seminar. I guess you can self-aggregate either upstairs in the biostat or outside, because it is a nice day, to have continuing discussion. So please join the speakers. Okay, thank you very much.